This is an ultrasound system. This mother's unborn child.
Right, this is baby's body here. So here's the heart beating. You can see it flickering away there. And this is the baby's aorta. This is a big vessel, a big artery that comes up from the baby's body and hooks round and enters into the baby's heart. And if we now put this little transducer on, we can hear and see the baby's heart beat coming through the cord.
In its body cells, that unborn child carries genetic material, DNA, inherited from its mother and its father. These cells have been drawn from around the embryo. They've been supplied with nutrient to encourage them to grow. In the warmth of the incubator, the blood cells will grow and eventually divide. As these cells divide, the chromosomes reproduce also. The new cell will contain DNA with exactly the same code as the original cell. The blood cells are placed on a slide. Because the cells are almost transparent, they have to be stained to be seen. At the moment this slide was prepared, some of those blood cells would have been dividing. This scientist is searching for evidence, searching for chromosomes. These are the chromosomes from a cell about to divide. The machine needs help to distinguish one chromosome from another. With help, the machine sorts out the 46 human chromosomes with their genes that regulate our bodies. Many people, including our own Queen Victoria, have possessed defective DNA, damaged genes. Victoria passed the defect to her daughter Alice. Alice passed the damaged portion of DNA in the sex chromosome to her daughter Alex. Alex, in turn, passed the defect to Alexis, who, as a result, suffered from haemophilia, a blood disorder. One of our parents may carry a defective gene on one chromosome. If both parents carry the same defect on a chromosome, then the child has a 1 in 4 chance of inheriting 2 defective genes.
Sickle cell disease is a common inherited genetic disorder. By that I mean it's a disorder that runs in families. It's passed on through different generations of families.
The people in my family who had sickle cell, my mother and my father, they passed on the genes to me, and now I've passed on the genes to my 6 children. My 5 boys has got the sickle cell trait. Rebecca has got the sickle cell disease because her father also had sickle cell.
It's a disease caused by the production of an abnormal hemoglobin, and hemoglobin is the pigment in your red blood cells. The disease is inherited on the chromosomes. We all have 2 sets of instructions carried on chromosomes. Most individuals have 2 sets of instructions for making a hemoglobin called hemoglobin A. People with sickle cell disease have the instructions to make hemoglobin S on both their chromosomes.
It gives a lot of pain. We call it a crisis. I've got it in my wrists. I've also got it in my knees, especially in my eye, which is the right eye. It's very blurry. I cannot see out of it whatsoever, but I've got lots of pain. I have to drink lots of fluid. But when I'm getting a real crisis, I have to go into hospital.
What happens in the disease is that these cells go from a nice round shape into an abnormally rigid, sickled shape. Nice round red blood cells will go through the blood vessels very easily, whereas these sickled cells will get stuck.
My granddad, I think he died with sickle cell in 1974. My mother also passed away in 1969. She also died with sickle cell. Rebecca's father just passed away 3 months now, and he also died with sickle cell.
Genetic diseases are caused by damaged DNA. No one yet knows how to restore damaged DNA. These are a strain of bacteria called E. coli. Small numbers of E. coli have been taken from the culture plate to be grown in a flask of liquid broth. These bacteria may help us to understand how some genetic disease may be cured. Like all living things, bacteria cells contain lengths of DNA. When left in the warm with plenty of nutrient, a single cell of bacterium grows and then divides. Each cell contains an identical chromosome. Each cell of bacterium also contains small pieces of DNA that have nothing to do with the chromosomes. Those tiny loops of DNA are called plasmids.
Plasmids are small pieces of DNA which are found in most living organisms. Because they're so small, we, for quite a number of these plasmids, know their complete genetic code. And that allows us to cut the DNA at specific places so that we can stick into the plasmid foreign pieces of DNA from animals or plants.
These tiny bottles contain a layer of plasmid DNA, separated from the bacteria. Scientists know exactly the building blocks that make up the molecule of plasmid DNA. They also know what enzymes can attack the molecule to break open the plasmid ring. If an enzyme is used to open a space in the ring, then a new piece of DNA can be added. This is an example of genetic engineering. Genetic engineering has helped us to understand genetic disease. If we can find what is wrong with the DNA of people with genetic disease, then it may be possible to create the correct piece of DNA and then copy it inside the cells of bacteria. Genetic disease may be treated by genetic material.
Alice, Samantha, you both have cystic fibrosis, and of course you were born with CF, because cystic fibrosis is an inherited disease. Tell us a bit about what it's like having CF.
I spend about an hour in the morning, or about an hour and a half in the morning actually, and an hour in the evening, doing physiotherapy and nebulized antibiotics. And also I have to keep up my appetite, because I think as soon as you lose your appetite, your weight goes down. And also I try to exercise as often as possible, because I find that that also helps me clear my chest.
I'm finding it okay at the moment. I mean, I get quite tired. That's my main problem, is I get tired a lot and quite breathless. So I find that sort of getting up early in the morning, you know, to be at lecture at 9 o'clock. By lunchtime, I'm shattered and sort of want to go to bed for the afternoon.
I find when I go to clubs or discos, everyone's on the dance floor dancing all night, and I can only last about one. I'm sitting out, and everyone else is up there. But I think as long as I can get out, that's better than sitting at home at all.
Yeah.
But I do find that smoke's a problem in clubs and discos,
and pubs and in closed areas especially.
But if you didn't go, you can't really go out where there isn't any smoke, and if you didn't, you just stay in. So there's nothing really we can do about that.
Does it affect you at work?
Well, at the moment, I'm doing voluntary work at a school because I find that I had trouble with employees where time off was concerned into hospital, and that's why I eventually gave up work to do voluntary work part-time. So I don't feel any pressurised to go into work. I can always put my treatment first, which I think is most important.
Cystic fibrosis is caused by damaged DNA on chromosome 7. This machine is extracting DNA as part of a search for damage in the DNA molecule. That huge molecule of DNA can be cut into lengths by using enzymes that attack the DNA at certain places. The blue liquid contains lengths of DNA colored with dye. In an electric field, those pieces of DNA move different distances. It's a way of checking for disease. It's also a way of searching for the particular piece of genetic damage that causes genetic disease.
We're very keen that people with CF should keep themselves as well as possible with the new treatments that are coming along. What do you think about gene therapy as a possible form of treatment?
Yeah, hopefully it'll mean great improvements in treatment for sufferers of cystic fibrosis.
And when the gene does come about, what form will it take? An inhalation, or a nebulizer, or tablet form?
It's only the lungs that would be treated in the first instance. It'd probably be inhaled in by nebulizer or some sort of spray. And the general idea is that if it's going to work anywhere, it'll work more easily on the lungs than it will in the digestive system.
Cystic fibrosis is caused by inheriting two defective genes. Those two defective genes may cause severe problems. Gene therapy will provide replacement DNA carried by a virus into the lungs. By the end of the century, gene therapy may offer a better way of treating cystic fibrosis.
But one of the reasons why gene therapy is so much more exciting than antibiotics or enzymes is that you're using the normal human gene for the treatment. Having that gene doesn't have any side effects, or else it would have them on us. So that's one of the main reasons why everyone's so excited at the possibility of gene therapy.